right, we'll get started. Welcome everyone to this ACUR at ANU student presentation session. My name's Oliver and I'll be your session chair today. I'm from the ANU, and so I'd first like to begin by acknowledging and celebrating the first Australians on whose traditional lands we meet. For me, that is the Ngunnawal and Ngambri people of Canberra, and I'd like to pay my respects to elders past, present and emerging. I'd also like to acknowledge the various lands from which people presenting today are coming from as well. As I mentioned before, this is a student presentation session made up of 3 presentations, each being 10 minutes. And today will be judged by 2 judges. Firstly, Dr. Aishi Zengin, and secondly, Associate Professor Shauna Gall. Thank you very much to the judges for giving up your time and coming to watch the presentations today. We'll get started right away. So I'll invite our first presenter today, who is Andrew Quattrocchi, who will be presenting about Alzheimer's disease, but I'll leave him to speak more about it.
Thanks, Oliver. All right. My undergraduate research aims to investigate the pathogenesis of Alzheimer's disease, specifically the role of perivascular macrophages in the brain. Alzheimer's disease is the most common neurodegenerative disease and is projected to triple in disease burden by 2050. Yet still, there is no effective therapeutic intervention. When thinking about Alzheimer's disease, the peptide amyloid beta may come to mind, where its aggregation forms plaques. Or maybe you've even heard of the insoluble tau protein that causes neurofibrillary tangles. What you probably did not know is that dysfunction in the blood-brain barrier and vascular leak is also another primary pathology. The blood-brain barrier is a highly selective, semi-permeable border that protects the brain. It is composed of endothelial cells, vascular smooth muscle cells and pericytes, and is supported by the feet of astrocytes. But what we have become interested in is what is the role of the perivascular macrophage in this complex vascular unit. So, it is understood that the blood-brain barrier perturbs with age. However, as demonstrated by this fMRI, where the red and yellow corresponds to the amount of leaking tissue, at a much earlier age in Alzheimer's disease, there is a significant increase in vascular dysfunction and leak. This leak occurs as a result in the alteration of the blood-brain barrier. Therefore, we are interested in understanding the cells of the blood-brain barrier and how they contribute to this vascular leak. So, what is a perivascular macrophage? Perivascular macrophages are immune cells that are intimately associated with the endothelium. One of their foundational roles is to phagocytose potentially harmful substances, cleaning up the microenvironment of aggregated protein, such as the amyloid beta we just discussed. They also support the basement membrane of the blood-brain barrier, adding to its functional integrity. They present antigens, playing a key role in the immunity of the central nervous system. And finally, they fundamentally control the vasculature, where they remodel its growth, support its maturation, as well as modulate blood flow. However, what I have time for to look at is what is their role in modulating the permeability of the endothelium. My hypothesis is that perivascular macrophages play a key role in early blood-brain barrier dysfunction in Alzheimer's disease. To address this hypothesis, my first research question and what I'll be presenting today is, do perivascular macrophage populations change in Alzheimer's disease? So what I first did was I went to the literature and looked at data that was available from single-cell RNA sequencing in both mice and humans. I then conducted my own immunofluorescence and confocal microscopy study. So what is single-cell sequencing? Single-cell RNA sequencing provides you with gene expression data at the level of individual single cells. So what you need to do is you need to get your sample into a single-cell input. This allows you to precisely determine the different cell types and subtypes in your sample. The reason why this is so powerful is it reveals the heterogeneity within these subtypes rather than the gene expression of the tissue as a whole. So what I did was I looked at studies that had already grouped their single-cell data using microglia markers, all of markers which are also expressed on perivascular macrophages. I then did a differential gene expression analysis on known markers of PBMs. The first marker I did was CD206. And to orient ourselves, the y-axis is a p-value, so any icon under the dashed line is significant. The x-axis shows the average log fold change. So a log fold change of 0.5 is indicative of a doubling of that gene expression. So as we can see in both mice and humans, CD206 expressing macrophages increase. This was also mirrored in live one expressing PVMs, another specific marker. Finally, I looked at CD163, but the story wasn't here as clear. As we can see, there's a heterogeneous expression of CD163 positive perivascular macrophages. In the prefrontal cortex, we can actually see there is a decrease. However, in the entorhinal cortex, there is a log fold change between 1 to 2. The reason why this is so important is the entorhinal cortex is one of the first structures in the brain where Alzheimer's disease pathology begins, showing us that these profiles of PVMs is different across the brain in Alzheimer's. So then, in my own study, I used the APPS1 transgenic mice model. This mice has 2 plasmids inserted into it, one resulting in an elevated secretion of that human amyloid beta peptide we spoke about, and finally, the other results in the aggregation of plaques. I looked at the mice in 2 different ages. First at 6 months of age when we can see the formation of plaques, and then at 2 months of age when there are no plaques forming. Using confocal microscopy and immunofluorescence, I was able to show that in Alzheimer's disease, compared to wild type, at 6 months of age, there is a significant increase in perivascular macrophages. I then looked at the different areas of the brain where this accounted to. So I first looked in the hippocampus, and this trend was seen, where there was an increase in the PVMs in Alzheimer's disease compared to wild type. I then looked at in the cortex more generally, and again, this trend was mirrored. These results are not statistically significant because I only had an N of 3. So what my next step is to look at more mice. I then looked at the morphology of these cells. So where a circle, more circular structure equates to more of a 1, an elliptical structure equates to about a 0.5, and a more straight structure equates to a 0, using this equation. And what I found was that perivascular macrophages in Alzheimer's disease compared to wild type are significantly more circular. So what I next did was, I looked at, well, where is this occurring? So I looked at different types of vessels, because it is known that PVMs like to sit on larger vessels, specifically the arteries and the venules, compared to the microvasculature. What I found was that this increase in circularity can be accounted for by the perivascular macrophages located on the large vasculature. So, in Alzheimer's disease, compared to wild type, we see the perivascular macrophages have a more amoeboid retracted morphology compared to the more traditional elongated morphology seen in the wild type mice. I then looked at the mice at 2 months of age to see if I could see this increase in perivascular macrophages and potentially a change in morphology. Whilst COVID stopped this, I did get some preliminary findings. So what I found was that in these preliminary results, there is a significant increase in perivascular macrophages. The reason why this is so exciting is because it suggests at 2 months of age, before plaque formation, one of the earliest observable changes in a model of Alzheimer's disease is this remodeling of the neurovascular niche. So what is happening? I believe 3 things could be possibly happening. Firstly, the perivascular macrophages could be retracting their processes and becoming more amoeboid. It is known in the literature that when macrophages change in morphology, they become more pro-inflammatory. Therefore, they decrease their contact with the endothelium, which I believe is associated with an inflamed endothelium and vascular leak, overall adding to the pro-inflammatory profile of the niche. This is particularly important in PVMs as they are one of the biggest producers of reactive oxygen species within the brain, further adding to this pro-inflammatory phenotype and neurodegeneration. Secondly, immune cells from the periphery could be sequestered through the blood-brain barrier into the brain to assist with the phagocytosis of amyloid beta. This is well known in the literature. So some alteration in the endothelium allows for the infiltration of immune cells. These sequestered monocytes can take on the same phenotype as the resident cells, therefore adding to the morphology and increase. So this immune infiltration ultimately adds to the pro-inflammatory profile as these sequestered monocytes are traditionally pro-inflammatory cells. Thirdly, both could be occurring. So both the retraction of the processes and the sequestering of monocytes into the blood-brain barrier, the production of reactive oxygen species could all be equating to this pro-inflammatory phenotype. So, what's next? I first need to confirm these findings in my young mice, something that was stopped by COVID but will definitely continue soon. So my question is, how is this occurring and where are they coming from? I'll do that using two different techniques. First, I'll do immunofluorescence and flow cytometry with a panel of specific antibodies, and then I will conduct an in vivo immune infiltration study using labeled monocytes. Your search, our future. People with dementia are going to triple by 2050. Even today, it's more than likely that one of you knows someone that has been affected by Alzheimer's disease. The vascular biology program at the Centenary Institute is searching for what controls the integrity of the blood-brain barrier. Ultimately, so in the future, we may have an effective therapeutic option to help cure this devastating disease. And I'd like to say thank you. Thank you very much.
Thank you for that fantastic presentation, Andrew. So I'll now open up for questions from the judges, but before I do that, I'd like to invite any of our attendees who have questions to either post them in the chat or ask them in the Q&A, and I will be monitoring those forums and can then convey questions to Andrew after the judges are finished with their questions. So please feel free, judges, to unmute and ask any questions you like.
Sure, I'm happy to start. Thanks, Andrew, for the presentation. I really loved the graphics. I think it was really well presented, so well done. I have a bit of an interest in sex differences in cardiovascular disease in particular, and I wondered if you could just comment on whether your mice were male and female, and if you've considered any sex differences in your study.
Yeah, definitely. So all my mice are male, which is pretty standard for science. However, sex difference is a big thing. So on one of my collaborators in my lab is looking at another cell type, so parasites in the brain. So they've age matched male and female, and it seems quite universal. So that's my next step. So as I said, those first findings was n equals 3. So the next stage is to get 3 female mice and then try to see if I can go over there. So
great, thanks, Andrew.
I'd like to echo my co-judges' compliments. Your talk was very nice and very understandable. Congratulations on this body of work. This is completely out of my area of expertise, so I wanted to ask you about your finding about the retraction of the macrophage processes. So sure, if this is the case, is there any way to reverse that, or what can be done about that?
What can be done about that? That's a great question. So what traditionally studies do, and it's a bit brute force kind of what they do, is you knock the macrophages out. So they use a drug, claudinate, and that can kind of get rid of all the macrophages, the perivascular macrophages. The problem with that, it's a global knockout. So you start knocking out microglia and you start knocking out other cells before they go into this pro-inflammatory phenotype. So what they've been able to show is by knocking out the PVMs, you can reduce some sort of inflammation. However, it's not really refined. It's not a refined process. So in the future, you'd want some sort of targeted therapy that can kind of prevent this inflammation profile. So this retraction is this pro-inflammatory profile. So what some people think about, so if you believe senescence, that's like a kind of way, you could use senolytics to kind of reduce that. If you could use like antioxidants, it's all different ways. But one of the biggest problems with brain drugs is that it won't pass through the blood-brain barrier. So we've got this double kind of like, how do we deal with the drug getting through the blood-brain barrier? But then as well, if the blood-brain barrier is perturbing, maybe then that's the time. So it's quite a complex question. Not 100% sure how you would do that. But as I said, the 3 ways you would think about is a drug to reduce the inflammation profile. And then the next step you'd have to overcome is, well, we need to get it into the brain before the blood-brain barrier perturbs. So you need a targeted method to get it through the blood-brain barrier. So there are we have collaborators in Queensland that they're using the ultrasound to transiently open the blood-brain barrier. So they're using that in conjunction with known Alzheimer's disease antibodies for the amyloid beta. So if you could transiently open the blood-brain barrier, get a drug in that could help reduce the inflammation of your perivascular macrophages, then you might be looking at something that kind of works.
Fantastic. Are there any other questions from judges or from attendees?
I can ask some more questions if we want to, yeah?
Yeah, maybe
keep rolling? Sure.
Yeah, maybe just one more and keep it to a short answer.
I just wanted to talk a bit about the study design in terms of, so were you blinded to the condition of the mice and so on in terms of the quality of what you did?
Yes, so I was blinded. Fundamentally, it's quite hard to be blinded because you can see the plaques. um, I was blind. So in some mice, you can't see plaques. So there's different formations of plaques. So they're quite sticky proteins. So even if you don't have an antibody for that plaque, it will stick to other of your antibodies. So for most of my mice, I didn't know what they were, but unfortunately, you can see the plaques. So in 1 out of the 3, like I knew was the Alzheimer's one. But yes, we went through the correct blinding process, but it is fundamentally quite hard.
Thanks.
Fantastic. Well, thank you for that presentation, Andrew. I think that will bring us to the end of today's session. I'd like to thank both of our judges once again and all of our student presenters. Congratulations on such fantastic presentations. Great visuals, and they really simplified down super complex topics, so well done. The recording of this session will be made available online following the conference, and we hope to see you all at other sessions in the conference. Bye, everyone.
Well done.
Thank you very much.
Bye.